Topic 7 is the study of blood, serology. Blood is actually a really complicated mixture of different things. You can find cells, enzymes, proteins, and inorganic substances. Plasma is the liquid portion of the blood, and it's mainly made of water. There are red blood cells, which are called erythrocytes, white blood cells, which are leukocytes, and platelets. Those are the other of the four main parts of blood. Those are the solids that are suspended in the liquid plasma. Red blood cells carry oxygen from the lungs to the body cells, and carbon dioxide from the cells back to the lungs. If we look at the surface of the red blood cells, there are antigens. These are proteins, and those are responsible for blood type characteristics. There are more than 15 blood antigen systems that have been identified, but the ones that are used with blood typing are the ABO and RH systems. And an individual that has type A has A antigens on the red blood cells, B has B antigens, AB has both A and B, and O is like a 0, has neither A nor B. So the presence or absence of two antigens, A and B, determine the ABO blood type. This is kind of what it would look like. So if a person is type A, here would be their red blood cell, and let's say they have these types of antigens, the A antigens, on the surface of the red blood cell. So this is type A. A person who is type B, try and fit this in here, their red blood cell antigens look a little bit different. A person who is type AB has both of those types of antigens on the surface of the cell. And then a person who is type O doesn't have any of them. Another blood typing antigen that's used is the Rh factor. This is also known as the D antigen. People who have the D antigen are Rh positive. This is just another protein that's found on the surface of the cell. So since it's positive, let's just make it a little plus sign, like that. D antigens are Rh positive, so this is positive, and negative, again, don't have this. If they don't have the antigen, they're Rh negative. For every antigen, there's an antibody that will react with it to form clumps known as agglutination. If serum or blood liquid containing anti-B antibodies is added to red blood cells carrying the B antigen, having that on the surface, they're going to react, they're going to clump together in the process called agglutination. Serology is the study of these antigen and serum antibody reactions. The identity of each of the four ABO blood groups can be established by testing the blood with anti-A and anti-B sera containing antibodies. So the antigen-antibody reactions has been applied to immunoassay techniques. I remember, or hopefully you remember that I mentioned that last topic about one of the screening tests for toxicology. That's detection of drugs, of abuse in blood and urine. EMIT, which is the immunoassay technology, it's the acronym, is a screening, not a confirmatory test for drugs. So what we see here is two different situations. In this case, on the left-hand side, we have type A red blood cells, and they don't react with anti-B antibodies. so there's no clumps formed, there's no agglutination. But when you have type B blood cells here, they will fit exactly with the anti-B antibodies, and they'll clump together. This is the agglutination. Forensic scientists who are studying blood have to be ready to answer the following questions. Is it blood, first of all? From what species did that blood come? If the blood is human, can we associate it with a particular individual? So the first step to that is it blood? And the determination of that is done by a screening test, a color test. So there's a Kastle-Meyer color test that you saw on the first slide of this presentation. Hemoglobin causes a deep pink color, which you see right here. You see that in some of the crime scene shows. Then there's the luminol test that's used to search out latent or trace amounts of blood located at crime scenes. It produces light in a darkened area under UV light. It'll glow, as you see here. Once it's been determined that the stain is blood, the precipitin test will determine whether it's human or animal blood. The precipitin test usually uses anti-sera from rabbits that have been injected with the blood of a known animal to determine the species origin of a blood stain. Then once it's determined that it's human, an effort has to be made to associate the stain with an individual. DNA analysis and DNA fingerprinting has brought us up to the point where we can associate a blood stain to a single individual. Blood typing will narrow it down, but it won't individualize it. So blood stains, when they were just blood typed with ABO blood typing, that's more of a class characteristic. It narrows down the number of people to which that blood stain might belong. But DNA technology gives us individualization. It's an individual characteristic. So we can associate those blood samples to a single individual. When we're looking at blood typing, we're looking at genes that are located on chromosomes. The different forms of genes are known as alleles. Paternity testing before DNA fingerprinting has involved blood typing, along with blood factors other than ABO, but mainly the ABO blood typing system. Now they use DNA testing, and that can establish paternity beyond 99% accuracy. When we look at the different genes that are determining blood type, we have two different words that we use. Homozygous is when both alleles are the same for that particular blood type. Heterozygous is when the two blood type alleles are different. We can also use the pluses and the minuses for this. So homozygous would be either 2 pluses or, whoops, 2 minuses. Heterozygous would be 1 plus and 1 minus for those alleles. Another thing we look at is the words genotype. Which is what you see up here. What do the genes look like? The phenotype is the physical characteristic, so the actual blood type, A, B, O, and positive or negative. That's the physical characteristics.